So today we'll be talking about mass spectrometric analysis of integral membrane proteins. So before we start, just to say that what we're going to cover today, there are some misconceptions about membrane protein mass spectrometry, so we will address those first. I'm then going to cover a number of techniques. There are a wide variety of techniques that you can use to analyze membrane proteins, and then we'll look at some applications of these techniques. So we'll make a start with membrane protein facts. If you work on membrane proteins, I apologize, you already know this. So membrane proteins are on the cell surface in general. They are receptors, transporters, enzymes, variety of different functions also involved in cell surface recognition, and they represent up to 30% of the human genome. They are very important. However, they are underrepresented in the protein data bank, and the reason for that you'll be familiar with. They are also extremely important drug targets. About half of all drug targets are G protein coupled receptors. They're very difficult to work on, as you probably know. They are generally low abundance. You need your protein to be highly expressed in order to do crystallography on your protein. That's quite difficult. They're difficult to express in high abundance, difficult to crystallize. The normal way of working with membrane proteins is to solubilize them with detergents. There are other ways of solubilizing membrane proteins. And here is the problem. Detergents in general are not compatible with mass spectrometry. They're very powerful ion suppressants. A lot of mass spec labs will not let you put sample with detergent anywhere near the mass spec. However, and this is the purpose of the talk today, all membrane proteins are amenable to all mass spectrometry analysis methods, and we'll discuss that as we go through. So some myths is, the first one is that you can't analyze membrane proteins by mass spec. That is patently untrue. We have been working on membrane proteins here at CMD for over a decade, and everything is accessible to mass spec. People will tell you the membrane protein won't fly. That's not correct. I will show you lots of spectra today where they do fly. People will claim the membrane protein sticks to the column. That is also not true. And also there is a general perception in the field that you need specialized equipment to do this type of analysis. So you need ion mobility, you have to have an orbit trap, you need nano electrospray, you need specially modified quadrupoles. This is not true. You can do all of this analysis on standard equipment. So, all membrane proteins are amenable to all mass spec analysis methods. This slide shows you some of the methods you can use for protein analysis. I've grouped them into three. So we have this kind of bottom-up tandem MS-MS methods here. Oops, going too far. We have top-down intact mass methods under denaturing conditions, and native intact mass methods. And the task for this analyses to work is to remove the detergent. So for MS-MS methods, the normal way that would be done would be through chloroform methanol partitioning. It can also be done by gel electrophoresis, and you also have the option of using labile mass spec friendly detergents. For intact mass analysis, we can use precipitation as a way of removing the detergent. We can then do LC-MS, or we can do direct infusion, and we'll discuss direct analysis by Methanol LC-MS. And the next most important method is direct infusion, where this is done under native conditions, and the detergent is still present, but it is removed by a process called collisional activation, which I'll discuss. Okay, so chloroform water methanol partitioning, this was developed because when people started doing proteomics, what they realized very soon was that membrane proteins, which are some of the most interesting proteins, are very poorly represented in a 2D gel proteomics experiment. And so what people started doing was to enrich for membrane proteins, and this is done using chloroform water methanol extraction. So it's a very simple technique, and I don't know if you can see this here, but there is an interface in between the chloroform methanol and the water, and at that interface, this is where your membrane proteins will concentrate at this interface, and you can then go on to do your proteomics experiment. However, if you use any type of enrichment in this type of experiment, it's absolutely essential to have some type of stable isotope labeling, because you need to quantitate, and if you have an enrichment, you can get differential enrichment from one sample to the next. So it's very important that you have something to enable accurate quantitation, and that's normally done by stable isotopes. Another method of dealing with membrane proteins is gel electrophoresis. So you can, in gel electrophoresis experiment, you would digest your protein, and so you would run your protein on SDS page gel. So the SDS, it would be about 1% in the gel, it'd be about 1% or 0.1% SDS. This is quite a powerful detergent. And as you go through the digestion protocol, that detergent is washed away. So the digest and the LC-MS is essentially unaffected by the presence of the original detergent you had and also the SDS. So this method works. It works pretty well, actually. We've used it a lot here at the CMD. The MS-MS coverage is not as high as it would be for soluble proteins. We covered the reasons for that in an earlier lecture. But all you need to compensate for that is you just require more protein, or you do slightly better MS-MS. Your MS-MS is more sensitive. And in our lab, I've said here a figure of 95%. It's actually much higher than that. We almost always succeed in identifying membrane proteins this way. I mentioned the use of detergents. So the detergent is not, normal detergents are not mass spec compatible. However, there are a couple of commercially available detergents which you can use to extract your membrane protein, and then you deactivate the detergent. You can either do it, in this case, through heat inactivation. So this causes dissociation of the detergent molecule, and it's no longer has the ion suppressant effect. Or you can do it with Rapogest. You can do this by low pH. And these methods have been widely used. I guess the first method that used intact mass measurement for membrane proteins is this one. So this was developed quite a long time ago, over 20 years ago now, and it's basically used as you need to precipitate the protein, and then you can perform very nice LC-MS using what I would describe as aggressive solvents. So the solvent system used here is a gradient of chloroform and neat formic acid. These are not really the solvents you particularly want to have in your mass spec lab. They're quite, I would say, hazardous. You need rather a lot of material, so this was done on quite abundant membrane proteins like bacteriorhodopsin. So you need quite a lot of protein. One thing that can happen is in the presence of these solvents, you can get methylation. And I would say this method is very seldom, if ever, used, but it was one of the first methods for doing this. I mentioned that there are different ways of solubilizing membrane proteins without detergent. So here's some example of some mass spectra. So these ones here, this is dodecamaltoside. This is a standard detergent that's used in membrane protein extraction and analysis. Here's another one. Sorry, my pointer is not working properly. So you can solubilize proteins in the presence of an amphipol. So you can obtain spectra that way. And also you have these disk-based methods. So this is an example of using a nanodisk to extract your protein, and then you can analyze that by mass spec. Here's another example of similar idea. This is solubilizing a membrane protein in the presence of a bicell. There are some issues with using these disk technologies. One of them is you are not going to get very distinct spectra, because you can't really control the number of lipids that will be incorporated into the nanodisk. So you don't get a single mass, you get a distribution of masses. So your sample is quite heterogeneous, and this can be a problem when you're trying to obtain spectra. This is a method that you may not be familiar with. So this is a kind of hybrid method. It's a specialized ionization technique called Lilbid. And how it works is you generate a spray of droplets containing your protein, and they are uncharged. Okay, so you generate a neutral spray of droplets. So it's not like electrospray, where these droplets are charged. They're neutral droplets. And what you do is you activate the droplet with the aid of a laser. And when the laser hits that droplet, it ablates the liquid, and it also imparts a charge onto the analyte. It can be either negative or positive. You can only measure one at a time. But you imagine you get a positively charged analyte in here, and then it just passes through a normal ion optic system, and it reaches the detector, and you get your signal. The reason I'm telling you about Lilbid is it will tolerate detergents. Here's an example of Lilbid which is being used for analysis of nanodiscs. There's only one lab. This is developed in Germany. There's only one lab that operates this, but it is quite a nice method, and it's something you should be aware of. So here at CMD, we have been working on intact membrane proteins for about 12 years now. This is one of the very first spectra that we obtained. This is the protein. It's a membrane protein. It's ABC transporter, ABCG2. And how we did this is we precipitated the protein, and then we resolubilized the protein in 100% formic acid. It works, but you do need rather a lot of protein. And the problem with any kind of precipitation technique is you need to get the protein back into solution. And in at least 50% of cases, this doesn't happen. So it's not a reliable technique. You need a lot of protein to do it. And personally, I would not really recommend it now. The reason we don't use that technique anymore is we developed a new method, and this is an LC-MS method based on the use of methanol. Normally, for LC-MS, we would be using acetonitrile. We discovered that actually it's not really possible to do LC-MS analysis of membrane proteins in the presence of acetonitrile, but it is possible in the presence of methanol. So how this method works is we have a fairly weakly binding column, a C3 column, so our protein will interact with that column, but not particularly strongly. We have a mobile phase, which is water and methanol. We run that over a medium length gradient, and what the idea is that we achieve chromatographic separation of the protein and the detergent. So the idea is we start with a protein detergent mixture, we separate those chromatographically so that the protein will be infused into the mass spectrometer at a different time from the detergent in our LC run. So we found this method is compatible with different types of detergents and also different lipids. And interestingly, it's also compatible with polyethylene glycol. Now, this is another ion suppressant, and very fortuitously, it also enables separation from that, which is very useful because it means that polyethylene glycol is very often used in your crystallization liquor, and it's possible to take a crystal, shoot the crystal in your X-ray source, get your structure, and then from the same crystal you can obtain a mass spectrum, so you know exactly what it was that you shot in your crystallography experiment. And also we can handle a fairly small amount of protein. That's about the same amount of protein we would use in the standard intact mass analysis. So here's an example of this method being used. I'm showing you here the total ion chromatograms. So this is where the aim is to separate our membrane protein from the detergent on the chromatographic column, and you can see we've succeeded in doing this. I've got 6 different examples here, 3 different detergents and 6 proteins, and they're all separated by roughly 2 minutes in the chromatogram. And then this peak here, this second peak, this is your protein eluting off the column, and this is a standard MZ spectra, and this will deconvolute and give you your structural information. I mentioned that the method can also be used for separating protein from polyethylene glycol. Here's the example. Here the separation is going the other way around. So the first total ion chromatograms I showed you, the detergent was eluting first and the protein was eluting afterwards. In this case, we have a protein USP8, which is contaminated with PEG 600. And the elution here is the protein is eluting first, and then the PEG is eluting afterwards. We have not achieved complete baseline to baseline resolution, but nevertheless, this is good enough, as you can see, to give us a good spectrum here, which will deconvolute nicely. And this is our PEG spectrum, which of course will not deconvolute because it's singly charged. So another method of removing detergent is to keep your protein solubilized in detergent, and it's possible by direct infusion to spray that directly into the mass spectrometer. Now, if you do this, normally what you will see is that you will not see the protein, you will see clusters of detergent, you'll see a big hump where your protein ought to be, but all you will see is detergent peaks. But what you can do is you can apply collisional activation. So you can, you have a collision cell in your mass spectrometer. If it's a quadrupole, it will have a collision cell. In that collision cell, you'll have a collision gas. It can be nitrogen or it can be argon. And you apply an accelerating voltage. And when you accelerate your protein through the collision cell, it collides with these gas molecules. Collisional activation means that the detergent that is associated with your protein is stripped away. And as you do this, your protein peaks will appear in the spectrum. So we do that here on this instrument. This is how it works. Our collision gas is argon. We apply this collision energy. And what happens, as I've said, is you get neutral loss of the detergent molecules. There is another thing which is possibly less desirable, is you get partial disruption of oligomeric states. Now, if you're interested in oligomeric states for native mass spec analysis, you have to bear this in mind. You don't get total disruption, but you do get partial disruption, and that makes the data analysis a little bit tricky. Okay, so if you're working on membrane protein, why should you bother with mass spec? What can it tell you? Well, actually, it can tell you quite a lot. I think the most important thing if you're working on a membrane protein is actually, is it the right protein? That may sound quite trivial, but I'll show you examples where actually it isn't. You must make sure that you are working on the right protein. Post-translation modifications are quite common for membrane proteins. Lipid modifications are very common. Also, because you're working on proteins that are membrane associated, glycosylation is very common. You can determine the covalent structure of your protein. From native mass spec, you can work out whether you have a native conformation or not. Obviously, you want to know if it's a complex, you want to know, do you have the complex and what is its stoichiometry? Another feature of membrane proteins is they very often have lipids either loosely or quite tightly associated with them. So you're going to want to know what these lipids are. Are there any other natural binders, any other natural ligands binding to your protein? Obviously, you're going to be interested in the mechanism of action, and native mass spec can tell you something about that. And finally, if you're interested in screening for drugs, you want to know which compound is interacting with your protein. You can also do that by native mass spectrometry. So, first question, do I have the right protein? It is not a trivial question. It's a very pertinent question. And I want to show you a piece of ancient history. This is something I worked on many years ago. This was for my PhD thesis, and I was working on a protein, or I thought I was working on a protein called cecropin. This is an antimicrobial peptide which I extracted from this beast, which is a yellow fever mosquito. And I knew the molecular weight that it should be and ran it on a gel, and you can see from the gel I have in my immune group I have the correct molecular weight compared to the standard from the gel. So we have the correct molecular weight. I'm looking for something that's about 4 kilodaltons. I knew its biological activity, so I had a bioassay and it came up positive in the bioassay, so that's all good. And finally, it's inducible. I had an immune stimulus, and that would induce 200-fold induction of the protein. So I have the correct molecular weight from STS page, I have the correct biological activity, and I have this 200-fold induction. So I was very confident that we've got the protein. This is what we're expecting. This is the sequence from the DNA. And we purified this. We sent it off for Edmund sequencing, as then we didn't have mass spec available. And this is the sequence we got back. I'm just aligning it with the cecropin sequence. And as you can see, there is absolutely no homology whatsoever. It's a completely different protein. It has the same biological activity, it has the same inducibility, it has the same gel mobility, and it is a completely different protein. This is an important lesson. You can never identify a protein by STS page. Lots of people try and do this. You are gambling. Most of the time, you will be correct. A certain proportion of the time, it will be a completely different protein. If you rely on STS page mobility to identify your protein, you should bear in mind that the identity, if we analyze your gel, we can find protein which matches your target by MS/MS theoretically anywhere on the gel. So this is your predicted mobility for your target protein. It ought to be here. However, if you have proteolysis, which is very common, you will see proteolysis products of the same protein at a lower molecular weight on the gel. If it's a membrane protein, all membrane proteins run faster than predicted on STS page. So you'll see it will be lower than you predict on the gel. If it's a glycoprotein, the opposite thing will happen. Your glycoprotein will run more slowly on the gel, so it'll have an apparently higher molecular weight. Of course, your protein could form multimers, so it'd be higher still on the gel. And it's also possible that you can have a read-through mutant of your target protein. So it's the correct protein, but it has the wrong mass because there is a mutation in the stop codon, and it reads through. You have the same protein with a larger than predicted mass. So basically, wherever your protein, your target protein can actually appear anywhere on your gel lane. Here's an example of a purification where someone was looking for this protein. This is KBTBD6A. That was a target. The MSMS, when it came back, it was a homolog, KBTD7A. So it's not the same protein. Similar molecular weight, different sequence. Here in CMD, we do lots of in-gel digest analyses. This is typical. So this is a month's worth of samples. It's over 300 samples. Most of them are membrane proteins. And as you can see from this analysis, only about 2/3 of them are the correct target protein. About a quarter of them are host protein. That's what you would expect. And a certain proportion are actually the wrong target. I wouldn't be surprised by that. When you're working on membrane proteins, it's very easy to mistake the protein, your target protein, for either a host protein or something else. Some labs will rely on reporters. So you may have something like here, you may have green fluorescent protein, and the assumption is if my protein is green, if my purification remains green, then I must have my target protein. That's not the case. I'm showing you two examples of analyses we've done here, like this one in the middle. This is the target protein. Well, they're all the same target. And here we have the presence of the green fluorescent protein, but the target is not present. So what's happened here is the target has been lost from the construct, but we still have GFP. Lots of labs will rely on Western blot. I would never rely on a Western blot. There are very good reasons for this. The first one is that I've put a figure here of 50% of commercial antibodies are non-specific. It may be higher than that. Bear in mind that your antibodies are validated by Western blot. Your Western blot is validated by SDS page mobility, and I've just said you cannot rely on SDS page mobility. So relying on a Western blot to identify your protein is, in my opinion, the height of recklessness. I'll give an example of how we can use membrane protein analysis here at CMD. We do high throughput test expression. This is an example of a 96-well 1 mil high throughput test expression for membrane proteins. So we have a range of different constructs for the membrane proteins of interest. And we have here in this first column, this is the yield of protein as measured by SDS page Kamasi blue staining. So here, this is high expressor, medium expressor, and low expressor, or in this case, no expression. For this range of constructs, we have an expected mass from the sequence. We can use intact mass to observe the mass, and then we compare the expected versus compared to the expected mass compared to the observed mass, and we have this delta mass here, and these delta masses are either going to be small or they're going to be a modification that we would expect. So this one is actually minus 98 daltons, and this is equivalent to loss of the N-terminal methionine and acetylation. The other thing we get on here is we can see the relative abundance. So we've got this figure here for the relative abundance in the mass spec, and we can use this to select the test expression for scale-up. So we can identify membrane proteins, we can check that we have the correct construct, we can see if there are any post-translation modifications, And we can do this, I would describe, in medium throughput. So this analysis was done over 20 hours. Here's an example of a post-translational modification. This is for an ion channel. This is SLC30A8. So this figure here, this corresponds to the expected mass, and we have a mass transition here of 239. This is actually 238, and this modification here is palmitoylation. It's extremely common. When we did this analysis, the postdoc who was working on SLC30, they said, I suggested let's have a look in UniProt and see what modifications are known for SLC30. So we had a look in UniProt, and palmitoylation wasn't there. So the postdoc got quite upset and said, well, why isn't it there? Well, the answer to that is you've just discovered it. Most labs don't do intact mass analysis of membrane proteins, so the palmitoylation is not known. So don't expect UniProt to necessarily know what the post-translation modifications are. This is the first description of palmitoylation for SLC30. Here's another membrane protein. PKD2 is a fairly large cell-to-cell matrix protein, and it is glycosylated. So you can see here we have multiple glycosylation states. Very often you wouldn't see this because you can see there is a lot of polydispersity in the sample, and normally you would treat with endo F to deglycosylate the protein. But of course, if you do that, you'll get your signal for the protein, but you won't see the glycosylation. It's quite nice to see the glycosylation. There are at least 5 glycosylation sites in PKD2, and you can see here this is glycosylation state 5, where all 5 are occupied. Most of them are in glycosylation state 4. Here's state 3, 2, and 1. And these mass separations here would correspond to individual sugars. This is PKD2 again. If you watch the structural mass spectrometry presentation, we went through how this was done. And this is an example of using mass spectrometry to determine whether this potential disulfide bond is real or not. So from the crystal structure, we knew that these 2 cysteines were in close proximity. The question is, are they disulfide bonded or not? We used differential MS-MS analysis to determine, actually, no, they're not. These cysteines are free, unlinked. Here's the other way of doing this, where we can use the charge state to determine that the PKD2 is actually, in the denaturing conditions, it's fully linear, which means it does not have any disulfide bonds. Here's an ion channel. This is a potassium transporter, KCNK2. This has rather a complex structure from its intact mass. It is a dimer. It's a covalent dimer, and because it's a dimer, it has 2 N-termini and 2 C-termini, and what you'll see in this rather complex spectrum is truncation at one or both of those termini. So you can see from this, you can determine what these truncations actually are. So here's an example now of a native analysis of another membrane protein. This is SLC14. We've used this technique I described of collisional activation. There's quite a lot going on in this spectrum, but there's an awful lot of information you can get. Essentially, this spectrum, there are 3 separate ion distributions. 1, 2, 3. The one in the middle, this is the native structure. From the charge distribution, we can determine that all of these peaks correspond to a trimer, SLC14, and we have what we would like to do is like to fit a Gaussian distribution to the trimer charge envelope. But what you discover is actually you cannot do that. The only way you can fit Gaussian curves to this trimer charge envelope is you have to fit 3. And what that tells you is that SLC14 has 3 stable conformers for the trimer. You can also see that there are these peaks here. These minor peaks are mass additions. Because we're in the native state, they correspond to non-covalent interaction, and we can see that the trimer is binding 3 different species, 243 Daltons, 278 Daltons, and 148 Daltons. I don't know what those species are. They may well be lipids. These other distributions, these are artifacts of the collisional activation process. So as well as stripping away detergent molecules, you can also strip away one of the multimer subunits. And that's what's happened here. And we know that this has happened because we have this unusual charging. So the charging here is unusually low. And what we're looking at is we're looking at the trimer that has lost one subunit, and it's lost more charge than it would normally get in the native state. And this is what's happened to it. This is the ejected monomer, and this has higher than normal charge. So this distribution here, which I've labeled in red, and this distribution here, which I've labeled in red, structurally, these don't really contain any information, and you should ignore these. Okay, these are artifacts of this collisional activation process. The biological data is here in this central distribution. I'll show you another one now. This is for another potassium transporter casing, K3. This forms a dimer. We have a similar situation here. You cannot fit a single Gaussian distribution. I have fitted 3 Gaussian distributions. So again, the dimer has 3 solution conformations. That's of interest because it's a transporter, and you want to know, well, how this transporter actually work. Well, you would expect that it would have different conformational states in order to allow your ions to pass through the channel, and that's being reflected here in these 3 different conformations, which you can see. So again, we have some post-translation modifications. This one, these are non-covalent modifications. So there's 203 Dalton PTM 1038. Again, they're probably lipids, but these modifications do not occur on the dimer or the trimer. And what that means, so they don't occur on the monomer. So what that means is these modifications are actually essential for the formation of the dimer. And again, we have these non-native structures. We have the dimer stripped of one subunit and depleted in charge, and then we have the ejected monomer, which has an excessive charge. And these are non-native structures. So the red are non-native, the green is native. I just want to say one more thing about instruments. So the consensus in native mass spectrometry is that you need to use an Orbitrap in order to do this type of work. The argument being that the Orbitrap will give you the highest resolution, and you need that high resolution in order to identify ligands, in order to identify potential drug binding events. Well, you do need that resolution, that's absolutely true. Interestingly, though, we've got a comparison here of the same protein. This is SLC14. This is the analysis done with an Orbitrap. this is specifically designed for this type of work, and this is an analysis done on a standard QTOF instrument. And what you can actually see is the resolution is higher on the QTOF. Not because there's anything special about the QTOF, it's because of the desolvation conditions that are being used. And you can see some of the same ligands, and you can actually see additional ligands with the QTOF. So I would recommend a QTOF for this type of work, personally. Here's another example of a membrane protein. It's a DPAG-T, and here we are seeing, I've got a comparison between the denatured intact mass spectrum for DPAG-T and a native spectrum for DPAG-T. And you can see that the native, we have mass addition. Mass addition is 774. Interestingly, you can see that this mass addition occurs more than once. So we have this addition of 1 molecule to 3 molecules on the same target protein. What that means is that this is a non-specific interaction, and that can be very useful to know that it's not going to be, maybe not biologically relevant, it's certainly not going to be a useful drug target because it's non-specifically binding. Here's another example of binding to a membrane protein. So KCNK10 is a covalent dimer. It has this rather complicated spectra because there are truncations. There's truncations from the homo and the heterodimer. So effectively, you have 5 peaks representing this dimer. This is a denatured, deconvoluted spectrum. If we now look at the deconvoluted spectrum from the native analysis, instead of having 5 peaks, we have 10 peaks. And those 10 peaks represent an addition of a non-covalent binder onto each of the 5 peaks we have here. So we have the same mass transition happening, it's 402 on all of these peaks. And what that tells us is, in this native spectrum, we have what I would describe as a fortuitous ligand binding. It's 403 Dalton. I don't know what that ligand is, but it'd be very interesting to do some more work on that. Finally, I want to show you this data from KCNK10. I described a couple of weeks ago how we do this. So this is a dynamic charge radius plot. So we are taking the charge state, we are calculating the charge radius, and then plotting that with normalized intensity. And what we have is, remember, this is produced by collisional activation, so we have this charge reduced monomer from the dimer, but we also have 1, 2, 3, 4 peaks in this charge radius plot. And what that tells us is that KCNK10 has 4 stable conformations. And this is known. KCNK10 it's a transporter, and in order to work, it needs to change its conformation. And it's known from crystallography that these conformational changes are actually quite dramatic. But you are seeing this here in the native spectrum. So I'm going to wind up now and just summarize. So I hope I've been able to demonstrate this afternoon that membrane proteins are amenable to mass spec, all mass spec methods. We've discussed different ways of removing detergent, and this is going to be done by SDS Page and Inundell digestion, by Methanol LC-MS, or by collisional activation for native mass spec. The mass spec, it will yield identity and structure. I would say this is essential to know this if you're working on a membrane protein, but it also yields additional information. It gives information on activity and conformation, stoichiometry for complexes, any natural ligands, fortuitous ligands, and also drug binding. So we use mass spec very heavily for membrane protein analysis here at CMD, and we do that, as I hope I've shown you, from the very start of the expression pipeline. And you can use mass spec for membrane proteins to inform and direct experimental decisions at the bench, and I believe this is one of the best ways to accelerate your research. And I would say if you're working on membrane proteins, it's suddenly become a very competitive field, and my advice is use the mass spec and use it to work smarter rather than working harder. That's all I'm going to say. That's all the seminars for this term. Next term, beginning on the 19th of January, we'll be talking about targeted phosphorylation analysis, and I'll be very happy to answer any questions you've got.